So our next presenter is Miguel Correia, and he actually got his PhD in 2015 from the University of Algarve, and he's now a postdoctoral researcher at the University of Algarve in Portugal. And basically his research interests include population assessment, ecology, biodiversity and conservation. And in his study, he focuses on threat assessment, population dynamics and mitigation actions. And today he will present one of his recently published papers, A Crown for Each Monarch, a distinguishable pattern using the photo idea. Miguel, we are very much looking forward to your talk.
Thank you, Olivia. It's a pleasure to be here. Being lucky enough to be a part of all the symposiums. So since the first one in Sweden, it's a pleasure to be here. I want to congratulate all the committee for this amazing symposium. I've sent a video, so if you can play it, I will very much appreciate it.
Okay. Miguel, I'll play a video. Please wait a moment.
Hello, my name is Miguel Correia. I'm very pleased to be here to talk about our study with photo identification with hippocampus. Our title of this presentation is A Crown for Each Monarch, a distinguishable pattern using photo ID. The authors of this study is myself, Daniel Antunes, Dr. José Pedro Andrade, and Dr. George Palmer. As you know, mark recapture studies need individual identification to allow proper estimate of population size, also to study several aspects of the fish biology and ecology, such as reproduction and survival. And also it looks at movement patterns and possible migration in these species. Most commonly used in fishes are external tags, which are designed to be implanted either intradermally or externally, as you can see in the pictures. This method is a bit expensive and highly invasive, and it might cause some skin lesions and bruises and also lead to occasional mortality. It usually comes in very big sizes and it's widely used in larger species. The plus side of this technique is that it's very visually easy to spot. In the case of the seahorses, external tags have also been used, particularly using necklaces. As in the case of any external tag, this tagging method is very invasive, and as is in the case of the necklaces, due to extensive handling. This may cause some lesions on the seahorse skin, and we also observe this with our tagged seahorses in our facilities, and you can see that is the case of the top left picture. We had this problem with our animals in our facilities, so we abandoned this type of tagging methodology. This tag also can be very hard to read in the wild, and particularly if these tags have attached some type of epithets or any type of dirt. Another technique that is also have been tested to be used as a tag is the use of this acoustic tag. This acoustic tag was used on the species Hippocampus rotulatus. These tags allow to collect information on movement patterns over a small period of time, and it can be identifiable even in low visibility conditions. And this is particularly important when you work inside of estuaries or lagoon systems with very low visibilities. However, this is very expensive and cannot be used over a long time period, and particularly due to the lifetime of the battery limitation. This process also can induce skin lesions in the seahorses, and they are still very bulky in size, although the size of the tags have been reducing over time. The most commonly used tagging method in seahorses is the use of elastomer ink. This ink reacts with UV lights and is injected in between the bony plates of each individual. You can use several color codes, and this color code will allow for individual identification in the wild. Although this technique is not as expensive as the external tags, they are still costly, and especially if you are considering assessing large populations of seahorses. As a cheaper alternative, it has been suggested the use of photo identification. It has proved to be successful in the identification of individuals in 2 seahorse species. So we have the case of Hippocampus reidii on top and Hippocampus gutulatus on the bottom. In the case of Hippocampus reidii, the morphological characteristic that allows individual identification is the shape of the top of the coronet. And in the case of Hippocampus gutulatus, the characteristic is the white spot pattern, as you can see is shown in the pictures. This method is marginally invasive, as it requires sometimes handling, but it can also be done with little or no handling at all. It is more time consuming if you think about all the images that have to be processed in the computer software. So that is the downside of this technique. In the Rio Formosa Lagoon, located in South Portugal, we have 2 seahorse species. We have Hippocampus gutulatus, which is the species on the left, and Hippocampus hippocampus, the species on the right. Since we have already identified an ID method for Hippocampus gutulatus, we wanted to test also if photo identification would work with Hippocampus hippocampus. As you can see from the 2 pictures, Hippocampus hippocampus doesn't have any very distinctive spot pattern, as is the case of Hippocampus gutulatus. So we had to look at other landmarks to be used for photo identification. In the case of the Hippocampus hippocampus, they have a coronet that is more proeminent, and so we wanted to try to test if this part of the seahorse body could be used as a distinguishable morphological characteristic. We have an ongoing breeding program in our field station. The field station is located in the heart of the Rio Formosa Lagoon. We do all sorts of experiments with seahorses since long. We started our breeding program that focused on both seahorse species that started since 2006 and is still ongoing. In our lab, we are able to make available every stage of both species life cycle. In the case of Hippocampus hippocampus, we also managed to have mature seahorses after 3 months old, which is very good. And we can breed them throughout the year. So this makes a very, very interesting species to use in experiments. So to test the validity of the use of photo identification in Hippocampus hippocampus, we used an F5 generation seahorses in 2 age classes. So we used young adults, age 4 months old, we use 49 young adults, 24 females and 25 males, and those are the size and weight characteristics. And we also use adults aged 15 months old, 45 adults with 23 females and 22 males with those sizes characteristics. So we made 2 photo sessions separated by 1 month between them for both age classes. We placed each seahorse in an 8-liter glass tank and placed the seahorse right next to the glass so it can be a better quality of photo. Each seahorse was always facing the same side, and we use a white plastic board to allow better contrast and to allow a better identification of the contour outline of the coronet. All the handling was done gently to avoid stress and skin abrasion. All photos were then downloaded to a folder in the computer and processed with the use of a computer software named i3S Contour. This software was designed to identify whales by looking at differences in contour of each individual tails. And so we thought that this could be a good software to have an application in Hippocampus hippocampus by focusing on the contour outline of each individual coronet. As per requirement of the software, we decided to use 2 different landmarks and to test 2 different contour outlines, which are shown here in the picture. So the first contour, or contour 1, went from the base of the eye spine to the 3rd trunk ring. And the contour 2 went from the tip of the snout to the 3rd trunk ring. This is an example of the procedure of this software, as we have to outline the contour of the coronet for each individual to be precise as possible. The software then creates a database with all the outlines, and using a special algorithm, it searches the database for matchings and provides a rank of possible matches, which are shown here in the picture. This can later be also validated by visual comparison to allow to add other possible distinctive characteristics to the process. Our main results show that there was a higher matching in contour 1 for adults when using contour 1 when comparing to young adults. This was also the case for contour 2. And this has probably to do with the size of the animals, which make it a bit harder to accurately outline each individual coronet if you are dealing with very small animals. Contour 2 provided higher first rank matches when compared with contour 1, which indicates the importance of the extension of the contour outline, so adding extra information to the software database. There were no statistical differences in gender in either contour experiments. So in conclusion, we reported that photo identification is a valid method for individual identification in Hippocampus hippocampus. This method is relatively cheap and non-invasive, as there is very little or to no handling at all. This is particularly relevant in more sensitive species, and especially when comparing to the existing alternatives. This method should be tested in other species to minimize stress and allow individual identification, and should be replicated in species in the wild. And I also highlight the fact that this method can be applied to photos in existing databases, such as iSeahorse or even iNaturalist. And overall, photo identification allows for a more accurate estimate of Hippocampus hippocampus in the wild, so it should be tested in the wild. As we proved in this research, the coronet shape seems to be an adequate characteristic to be distinguishable for individuals from 4 months old onwards. Studies have also reported that full ossification occurs in seahorses ages 1 month old onwards, which indicates that the coordinate shape does not change significantly after that time. Photo quality is very important, as is the correct positioning of the seahorse individuals. This is vital for an accurate outline of the contour of the coordinates when processing in the software database. And one of the possible challenges, besides the quality of the photos that I referred, can be the presence of skin filaments or other type of protuberances, which have been reported to change the size occasionally over time. And this also, of course, can impair the proper outline of the contour of the coronet. So we proved with this study that photo identification is a valid method for hippocampus. Now it's time to test this in the wild and also try to test this method without the species. Thank you very much for your time.
Thank you, Miguel, for this very interesting talk. I think this sounds like a great tool to have available to both do in the field, but also in the lab. For example, we always have the issue that we cannot really differentiate the fishes when we have several in the aquarium. So are there questions? Please just unmute yourself if you have one. Okay, then I ask a question. So what are now your ideas what do you do now with this identification tool in the field? What are your plans?
Yeah, so the idea is to try to assess seahorse population sizes and also fluctuations, because here in the Rio Formosa, we have very high impact of human activities. We also been having illegal trade targeting seahorses. And so, as you can imagine, this has a very big impact on the wild population. So what we want to do is to try to assess an accurate estimate of the size of the population to try and see if the trend is still a declining trend or if there's somewhat a recovery of the population over time.
Yeah, thank you. This sounds very important.
I think Natalie has a question.
Yeah, Natalie, please. You have to unmute yourself.
I did it. Hi, Miguel. Congratulations. I loved your work. I have a question about how you use your photos. How many photos you need to have a good accuracy of your study? Because I know E3S demands sometimes more than one picture to have a nice accuracy. And unfortunately, our technique with the cornet functions very well when the seahorses do not have skin filaments, but when they do have, that's not useful. So I loved your other work with the spots, then maybe we'll try here in Hippocampus rhydi. So how many pictures do you need to have a good accuracy?
That's very exciting, and of course, our work with Hippocampus was somehow what inspired with your own work with rhydi, so that was very useful. Of course, you have to have a fairly big size of individual to prove the accuracy of the system and the technique. We are lucky enough to be able to read, I would say, fairly good amount of seahorses. So we have access to hundreds of seahorses if we need to do the experiments with. But I would say that at least, to make it very, very robust, at least 50 would be the mark for having consistent data to achieve that. Of course, the skin filaments are very, very tricky. Also, Hippocampus might have skin filaments. It's not exclusive of Hippocampus guttulatus. And in that case, it will be a big challenge to look at the coordinate shape. But this is particularly good in species where skin filaments are almost absent or really absent. And it could be tested in other species, and it should be tested in other species. The spot pattern is also a good option. We had very good results with Hippocampus guttulatus. So yeah, you should go for it.
Thank you. I have another question about how many pictures, individual pictures you need to take to have a good result. One
Yeah,
is enough.
so yeah, it depends on the picture quality, and that's the biggest concern, is to have good quality with good definition. If it's a bit blurry, it makes the outline contour very difficult. And so that's a big challenge. And if you think that you're taking picture from the wild, that sometimes we have poor visibility or other constraints, it could be a challenge to look at having a really good quality picture. So I would suggest taking as many pictures as you can to assure that you have a good quality pictures in the midst. But in theory, one would be enough. If it's good quality enough, one would be enough. And the good of this method is that you can use photos that are already in databases and try to look for, I wouldn't say population sizes, but see if there's resightings over a specific location of the same individual. And with this tool, you can do that.
Thank you.
Thank you. I see another question from Nuno. Just unmute yourself.
Yeah, thank you. Very nice work, Miguel, as usual. Have you measured the plasticity during the development of the shape of the coronet? And a second, probably more stupid question, do the kids look like their fathers? Do you have some inheritance in the shape, or?
That's a very good question. We haven't looked into that. As for plasticity, we assumed with the previous work that says that ossification occurs after 1 month old, we assumed that the shape wouldn't change that much. And the fact is that we only took a picture or a small frame of the entire life cycle of the animal. So that could be a good experiment to do, to follow the growth of the seahorse and see if the shape of the coronet would change over time. I don't believe so, but it's an interesting study that could be done. And with regards of having the same coronet as the fathers, I don't think so. It changes a lot, and we can see that throughout our many generations that we had in captivity. Coronet shapes vary a lot, and I don't think there's direct correlation with the parents.
So thank you again, Miguel, for your talk.